But I have emailed people again today, and I was cranky this time. Usually I'm really nice, but I wasn't very nice today, emailing, asking them to fix Echo. So hopefully they'll come and fix Echo sometime today. Today we are going to finish. Ulrich, are you recording this?
Yeah.
Hopefully you weren't recording. I'll be hanging out under the
No.
Yes. Yesterday I was telling her about this not working. And I told her that you were recording lectures so that we could post online. And I told her what your name was, and she goes, Sir Ulrich von Lichtenstein.
Yeah.
What movie is that? A Knight's Tale. A Knight's Tale. Anyway, I was telling her I liked your name. It was really cool. She said, it reminded her of A Knight's Tale. Man, I love A Knight's Tale. It's super good. Okay, the plan from here on out. Your exam is going to be on, let me give you an exact date. Your exam is going to be on November 29th. Wednesday, November 29th.
Wednesday?
Oh, Wednesday is crazy, isn't it? Oh, Wednesday, November 29th.
He said Friday.
I lied. For sure, 100%. Wednesday the 29th. That will give us 2 to 3 days per chapter in here, which is going to be plenty of time. Today we're going to finish this chapter. We're probably not going to go into the next chapter because I'll have to talk to you for a minute after we finish. You're not in trouble. The room has cleaned out a little bit.
Really drunk.
Yes. Before we finish this, we need to go back because I forgot something. I completely forgot to talk about something. And I meant to forget about it for your first exam, but I forgot about it yesterday, or Monday. Monday. Okay. One thing I forgot to talk about was, this is important because we will see this in your exam, base hydrolysis of nucleotides. Nucleic acids, I should say. DNA, RNA. You put DNA or RNA in a perfect solution, pH 7. Both of them will slowly hydrolyze over time. But cloudy nature will break your nucleic acid bonds. What kind of bond is that? It is a 3' 5' phosphodiester bond. Over time, you're going to break these bonds. One of these hydrolyzes more readily than the other. Which one? RNA hydrolyzes more readily than DNA. Why does RNA hydrolyze more readily than DNA? Because of the only difference in RNA and DNA. The hydroxyl group at the 2' position. Why does this make a difference and does it make a large difference? The answer is it does make a large difference. And why? We're going to talk about that. In order to talk about that, I want to draw this a little differently. You'll see in your textbook a shorthand version of a polypeptide drawn like so. What does this mean? If you're looking at a polypeptide, you may see a letter up here. These letters stand for your bases. And your specific carbons are not labeled, but they're assumed to be carbon on your ribose. Your little check mark is your ribose. This is a polynucleotide. Carbon number 1 is labeled here. Carbon number 2 is not labeled. Carbon number 3 is labeled here. That's where your phosphodiester bridge occurs. Carbon number 4. Carbon number 5. Right. This is the end. So you will see this written shorthand in your textbook. You may see it on a standardized exam this way. You won't see it on our exams this way. But it is easy to show simple schemes, such as hydrolysis of a nucleic acid. Hydrolysis of a nucleic acid. Strong. Phosphate. Phosphodiester bridge. let's say, adenine. In an RNA, at our 2' position, we have an OH, right? DNA at our 2' position, we have hydrogen. It makes a large difference. Base hydrolysis. Why? Because in the presence of base, base can do base things. Extract a proton from this OH group, which would leave us with an electron-rich, highly charged oxyanion and a nice partially positive charge, electron-deficient nucleophile on our phosphate in fairly close proximity, so we get nucleophilic attack from our resulting oxyanion to our nucleophilic phosphorus on our phosphate anion. This results in cleavage of bond here, formation of an additional oxygen phosphate bond. And what are we left with? We're left with, we're left with a resulting and a ring structure. We'll also rearrange the cell. A resulting ring structure. Yes, and water moves it. At the 2', 3' position. And this can rearrange itself either way. You can cleave this bond, or it can cleave this bond. So you're left with a racemic mixture of phosphate, inorganic phosphate on your ribose sugar at the 2' or 3' position. This is the only way you're going to get inorganic phosphate at the 2' position. Right? If you cleave this bond here, your OH would now be at the 3' position, and your phosphate would be at the 2' position. So that's something that you're asked if you see a structure in which your phosphate on your nucleotide is at the 2' position. How do you get there? Base hydrolysis of RNA or base hydrolysis. The idea. Both of these are hydrolyzed in the presence of base.
Does it cleave on the right side of the oxygen or on the left on that top? Were you marking blue? Top one. No, the top picture.
This?
Yeah. Does it cleave on the right?
Oh, no, you're right. So you're right. These do. This was fine. And this. No, you're right. Thank you. And I don't have white. So, no, you're right. Cleavage occurs here. These are going to be left with an OH on here, resulting.
So in the diagram below, is it going to cleave at the left side?
No, I drew the diagram below right
Okay.
initially. Sorry, I prematurely started scratching things out. No, I drew this correct. I didn't do it. Cleavage occurs at this side. This was right initially. The main idea. Base hydrolysis does occur in both situations, even under the smallest presence of base, even at neutral pH conditions. Slowly, it occurs more readily in RNA than DNA. Why? Because you can imagine if this was not an OH, this was just an AG, it would be much harder to get any kind of attack on your phosphorus. Let's continue. Now let's finish, and then after we finish, I'll go through and I'll tell you exactly this is what it's going to be on your. These are the things you're going to need to know for these chapters on your exam. Pretty much what we talked about since your last. DNA. We are going to talk about secondary structure of DNA very briefly. Why? Because it really helps to explain cloning, recombinant DNA. Why we're working with circular DNA. Helps explain PCR, which are the main two difficult concepts of this. I know some of the things we talked about in the last class; those are new concepts, very various deviations in base pairing. Those are things you need to know. And those are things you probably haven't talked about before. But this is going to be kind of weird. Definitely something new, unless you've done it in a research lab. And before we talk about supercoiled DNA, a specific secondary structure of certain DNAs, we kind of need to describe plasmids. What are plasmids? Plasmids are circular DNA, extrachromosomal. So DNA outside of a nucleus, outside of a chromosome. We'll define chromatin in a second. Packaged DNA that's outside of its packaged storage form, basically. Exist to a very large degree, especially in bacteria, exist as circular DNA. This circular DNA can adopt specific secondary structures. It can coil, it can twist, it can writhe. So these plasmids are circular DNA. And twist, which is, I guess it can kind of, it's kind of hard to explain. Writhe. I could draw nice pictures for you, but I can't draw very well, so I'm not going to. If you look in your textbook, it shows you, both textbooks will show you differences between twisting and writhing. Twisting is kind of more like coiling. Writhing would be kind of like wringing out a wet paper towel or a wet rag. You're wringing out something that's wet. That's a writhe. So twist is when DNA is coiling around itself, especially circular DNA. It's coiling around itself. Writhing is when that circular DNA starts to twist. If you've ever kind of coiled up an extension cord, like a thick gauge extension cord, you know that it's going to start doing weird things. Doesn't coil as easy as you think it would coil. It starts flopping all over the place, and if you don't twist it right as you're pulling it up, it's going to start writhing around on itself. Those are the types of things we're talking about. This can all be described. The degree in which this supercoiled DNA, this specific secondary structure exists, is described by a value L, which is called our linking number. And all it is the number of twist and rise. The number of twist and the number of rise in a circular set of DNA. And if it is a circle, DNA is circular and it is connected. You can imagine that this number doesn't change. If you twist 5 times and you writhe once, you're going to take a twist away. You writhe twice, you're going to take another twist away. So it doesn't really matter how many twists versus how many rise. That L is going to be the same, and all it is describing is the complexity of the secondary structure. And this is something that I think is kind of silly, but ACS thinks this is very important. So if you've ever taken ACS exam, this is good to know. Otherwise, I thought about it. Chromosomes. What do we mean when we're talking about chromosomes? We're talking about DNA as chromatin. Interphase cycle. During the interphase cycle, chromosomes, DNA exists as chromatin. Chromatin is a complex macromolecule. So at this point, chromatin does not exist just as DNA. Chromosomes do not exist simply as DNA. They exist as a complex macromolecule we call chromatin. And these consist of various proteins. These proteins can be labeled histones or non-histones. Histones make up the larger quantity of proteins involved in chromatin. Histones are like boring proteins. They don't really do anything. I guess they do stuff. They provide structural support and specifically packaging. You can imagine that there's a lot of DNA contained in chromatin and nuclei. It is packaged and stored in a very, very small place. And it is a very complex procedure, and the exact function, the exact mechanism of histones aren't known very well. But we do know that We may not be sure exactly how they package these, but they do package them. Whether it's wrapping, folding, whatever they do with the DNA. They take DNA, they package it in a way that doesn't damage it and cram everything into a very, very small space. Non-histones are a much smaller portion or percentage of the proteins involved in this macromolecule, but they are the more interesting ones. They do stuff. They're involved in things like DNA repair, replication, and expression. The topic that we're fixing to talk about, we're going to talk about two topics. We're going to talk about cloning, which is really weird and kind of freaks people out. I'm not talking about having a sheep and then having two sheep. That's not what I'm talking about. I'm talking about cloning. We're not there yet. We're not there. We won't go there. We're talking about cloning, recombinant DNA. You can obviously imagine recombined DNA. We're taking DNA, we're changing it, we're combining it, we're making something new. That's what we mean by cloning. And then we're going to talk about amplification. Right after that, they go hand in hand, cloning and amplification. And amplification, such as we're going to talk about one method called polymerase chain reaction, which is very important. This is why I don't talk about RNAs, DNA polymerase in class. Obviously, you need to know those things. Specifically, you need to know how DNA polymerase works. Why? Because that's what we're doing when we do PCR. Knowing how DNA polymerase doesn't help us that much by itself, because how can we manipulate that? PCR is how we can manipulate that. So we'll talk about that right afterwards. But this is a weird topic, and I expect questions. If you don't ask questions, I'm going to assume that you guys, one, you don't care, or which that may be the case, and that's fine. But it's going to be on your exam, so you have to care to some degree, right? But you're going to have questions, so you need to ask questions. I promise you. It's a weird topic. Before we get into cloning, we're going to talk about, restriction enzymes. What are restriction enzymes? Restriction enzymes are enzymes that cleave double-stranded DNA. And there are a lot of enzymes that can nick DNA. Nick DNA, but they can't cleave double-stranded DNA. What I mean by nick DNA, they can cut a single strand of DNA or a strand of RNA. They can't cleave double-stranded DNA or RNA. Those that can, we call restriction enzymes. Why? Because they were first identified in bacteria that basically chopped up invaders. Invading viruses, that's probably the initial function, the main function of restriction enzymes. Sure, we're taking advantage of them and using them in a different sense, but their role is to chop up DNA, chop up viral DNA, chop up invading DNA, so it can't replicate, so it can't do this. So they can cleave double-stranded DNA, and they can do this two ways. Restriction enzymes can do this two ways. They can have sticky end cleavage or blunt end cleavage. And sticky is one of my favorite words. Try saying sticky and not smiling when you're done. Try it. Sticky. Your leaves are, you giggle. It's funny. It's a funny word. Sticky end cleavage. You can imagine. So if we have a double strand of DNA, double strand of DNA going on over here. Sticky end cleavage would be cleaving a strand of DNA in a place that results in two sides of your DNA. You have a little bit of a lip or a hangover. You can imagine if you put a restriction enzyme and it cleaved this DNA, you didn't put, eventually what's it going to do? It's just going to recombine, right? So you cleave this DNA. Restriction enzymes are different because they nick both sides of double-stranded DNA, not just a nick in DNA. They nick both sides, so it cleaves double-stranded DNA. And you're left with two ends. You can imagine sticky end cleavage. Eventually, what's going to happen? These are going to come back together. We'll take advantage of that in a second. Blunt end cleavage. At this point, you probably don't need me to draw it because you get it. It's different in the fact that blunt end cleavage is a restriction enzyme that recognizes the exact same spot on both sides of a strand Innately, there's nothing different other than the site it recognizes. The only difference is the site it recognizes. And it's going to leave us with 2 strands of DNA. Like so. And what's going to happen to this? Without any help, it's not going to do anything, right? It's not going to do anything. Innately, you cleave it, it doesn't do anything. So these are really good. If you're wanting to chop up invading DNA, this is a great way to chop up invading DNA, right? Because it's not going to be able to do anything. So 2 types of restriction enzymes. You should be able to identify based on the target. If I tell you, here's 4 or 5 restriction enzymes, this is what they recognize and where they cleave, you should be able to identify what is it going to generate. Why? Because you know base pairing at this point, right? You know base pairing. So if I told you had a restriction enzyme, SMA1, it's a type 1 restriction enzyme. In your textbook, I'm not going to cover this in class, but in your textbook, you should read about type 1, type 2, and type 3 restriction enzymes and be able to identify the difference. So if you want to that. Be able to identify the difference. In 1 sentence is completely fine. Be able to identify in 1 sentence what's the difference between type 1, type 2, and type 3 restriction enzymes. We have a type 1 restriction enzyme that will cleave, recognizes CCC, G, and it will cleave in between CCC, GGG. Is that going to yield blunt or sticky cleavage? Blunt or sticky? How do you know? Because what? They match. What do you mean by they match? CCC corresponds to GGG. So what? So tell me more than just they match. I know that you understand what you're talking about, but if this was a short answer question and you told me because they match, it was 10 points, I'd be, he may know what he's talking about, +5. Tell me that you know exactly why this would be blunt in cleavage.
The other strand is cleaved.
Because the other strand, what?
The other strands cleave at the same spot.
The other strand will cleave at the same spot because you know how these base pair. And if this is CCC, so if I'm reading this way, right? I'm reading after a CCC and before a GGG, that's when we cleave. After a CCC, and if these correspond GGG, it's going to cleave in the exact same spot, right? So you know base pairing. And since one of the sides corresponds to the exact other side, it's going to yield blunt in cleavage. And that's something you should definitely be able to do for your exam. If you're given various types, various things, you should be able to identify, will this yield blunt in, or will this yield sticky? I promise you will see that on the So restriction enzymes can cleave blunt and can cleave sticky. How do we use restriction enzymes and plasmids as cloning vectors? And what do we mean when we say a cloning vector or a vector? When we say vectors, we're talking about isolated DNA. Remember, circular DNA. We're talking about this is a vector, a plasmid, a circular strand of DNA that we can use as a vehicle to change and carry that change into a host. So we have a strand of DNA. We talked about this. We hinted on this a long time ago when we talked about protein purification and overexpression, right? We hinted on this, and I told you there's some proteins you can manipulate your cell to overexpress that protein. This is how you do that. This is how you do that. You basically take a strand of DNA and you tell it what you want it to do. How do we tell it what we want it to do? So by far, the most common strands of DNA and cell lines that are used to do cloning, protein purification, and mutations. By the way, this is how we do site-directed mutagenesis as well. Anyway, to do all of these things, how do we do these? We do these using cloning vectors, PBR 322. And I don't mean pro bull riding. Anybody a bull rider? Any bull riders? No? You were just thinking that? I played soccer. Yeah. I played soccer with a guy in Abilene that was a bull rider, and that's what he did for real. And, it was crazy. If you've I've never done this, but I've been within 5 feet of other people doing it. It's nuts. people are crazy. I'd rather jump out of an airplane. Anybody jump out of an airplane? You jumped out of it? Was it fun? Terrifying? Don't act like you aren't scared. It's gotta be terrifying. I would rather jump, I won't do it either, but I would rather jump out of an airplane than get on the back of an angry bull. Anyway, these clone vectors, various E. coli. So when you think of E. coli, you think of, oh, pathogenic. Most E. coli is not pathogenic. Most E. coli is fairly harmless. Specific strands of E. coli and specific plasmids from non-pathogenic E. coli are the main vectors, the main cloning vectors used in cloning and mutagenesis. One of the very first, and this is one that's not really used much anymore. You can tell your textbook's really, really old when it's talking about these vectors. It's a great teaching method. It's not used a lot anymore. But one of the first vectors is PBR 322. One of the first vectors identified for cloning. PBR 322. A native vector to non-pathogenic bacteria that encodes for a few things. One of the things it encodes for is ampicillin resistance. One of the things it encodes for is tetracycline resistance, 2 antibiotic resistances. So you can imagine if you wanted to grow bacteria that expressed this plasmid, you could grow them in the presence of 2 bacteria, and they're the only things that are going to grow. Other plasmids don't have these two specific regions that encodes for production of something that makes them resistant to these 2 antibiotic. So you'll see in your textbook a few methods of how this works. You grow bacteria in the presence of ampicillin, tetracycline. These are the only bacteria that lives. You can mutate one of these away. And if you mutate one of these away, we do so by removing part of the DNA and inserting DNA. So if you want to change something, you've got to remove something and you've got to insert something. So if you look at, let's say, BAMH1. This is a restriction enzyme that yields sticky end cleavage. It cleaves, and here's a pseudospine. It says, oh, it cleaves right here. It actually cleaves in multiple. It cleaves in 4 places. Most of these restriction enzymes, if they're good restriction enzymes, if it's blunt-ended, it'll just cleave in one place, right? Most useful restriction enzymes are sticky end cleavage enzymes. Why? Because we want to cut something, we want to insert something. If we use a blunt end cleavage, all it's going to do is chop up the DNA in one spot, and we won't be able to recombine. What do we do with that? Anyway, so it is a sticky end cleavage. It's here, it's somewhere in here, somewhere around here. So what ends up happening is it removes a chunk of DNA. Removes a nice section of DNA. And we talked about before, I told you that DNA is not expensive, right? To generate DNA, generating polynucleotide strands is not expensive. It's not difficult. It requires a setup to do, and most labs aren't set up to do it. However, various vendors will do this. You can generate a strand of DNA to fit. Let's say we cleaved here and we wanted to generate 2 strands that would fit here, that would be complementary to those sticky ends and would contain, you can imagine, we would need, if we were generating, we would need something that's complementary here just to make it stick, right? We need something complementary here just to make it stick. We need something complementary here to make it stick. And then we would need good information. Good information. This is where my change is occurring. Good information that we're trying to splice into this gene. So we have a part of our 2 primers that are just designed to stick to what I cut. And then we have the middle. What's inserted in there is what I'm actually wanting to change. $7, $8, you can pay somebody to make these DNA, these primer strands, call them primer strands. What are they? They are single-stranded DNA strands that for $7, why would you do this? It's just not worth it. Pay somebody $7. You have to generate the sequences. You tell them exactly what you want. They generate it. You put in the solution. You do all this work. They make the DNA for you. You insert it. However, if we were going to do this, we're going to insert right here the one thing, even if we didn't make any other changes, the one thing that we know we changed is this is no longer resistant to tetracycline. This is no longer resistant to a certain bacteria. So in your textbook, it has a really cool picture of how you would go about making one change and then getting bacteria from this change. If you wanted bacteria that had this gene, how would you get it, right? Because you can't, if you grew bacteria after this change in the presence of both antibiotics, it's just going to kill what you made, right? Because what you made is no longer resistant to tetracycline. So it actually walks through some cool change. Some simple things, that's kind of difficult. Some simple things is you can make DNA that is resistant to certain antibiotics. So you could actually insert a gene that is resistant to a different antibiotic, and that makes it a lot easier. So the examples in your textbook are kind of cool, but they are kind of old school. Newer ways are much easier. I told you at the very beginning of class, not the very beginning, when we started talking about protein purification, the enzymes that I worked on for my PhD was to make it easier to purify. We actually encoded that not only do you produce this enzyme, you also produce this enzyme attached to another enzyme with covalent modification. It makes it easy. This is how these things are done. Site-directed mutagenesis. This is how these things are done. If you have a vector that produces your protein, if you want to do site-directed mutagenesis, this is where you do site-directed mutagenesis. You will mutate. You will take this. You will mutate one amino acid. Right? You'll mutate a certain sequence that encodes for a specific amino acid. And that's how you get your change. You grow these cells. So once we make a mutation, or once you make a change to your host DNA, your vector DNA, you grow this DNA up. And then how in the world do we get a lot of it? Once you make a change, and you're able to isolate that change, make a change, you're able to isolate that change, you have to get a measurable quantity of that changed material. Back in the day, this was a very, very tedious and a very, very slow process. Until the advent of polymerase chain reaction. I'm going to turn this sideways because it's a little easier. Kind of a little cartoon that goes through how polymerase chain reaction works and what it does. So we've modified our DNA. How do we modify DNA? Using restriction enzymes in a cloning vector, which is a plasmid, a useful plasmid that will work in a certain strand or bacteria. Once we get that bacteria with our new strand and we've isolated it, how do we extract that DNA? Extracting DNA from a single colony of bacteria is not very effective. Well, it is effective. You can get a small quantity that is not really, can't really work with it. But fortunately, polymerase chain reaction is able to amplify that by orders of magnitude, 6, 7 orders of magnitude. It's pretty easy to do, and it's very cheap. And this is one of the things that you can buy kits that do PCR, and they come with instructions. And honestly, a monkey could do it better than a human. Have you ever done PCR? Yeah. Has anybody ever done PCR? You've done PCR. So did you use a kit?
We had like a little set, wasn't an individual one though. So
Okay.
the professor gave out all the materials and told us what to do and said go for it.
Okay. You can buy these little kits, and it's literally a box. And I'll say you can do 20 PCR reactions with this box. It's like $200. And it's awesome because it literally gives you a step-by-step walkthrough of this is what you do. And if you change anything, it's funny because as a PhD chemist, as a graduate school, like going to graduate school, getting your PhD, you are trying to find ways to save money all the time because your professor doesn't want to give you money. They don't want to buy things, so you're trying to change. Oh, I can do this cheaper. I can do this. We don't have any. That's the main thing. We never have any of this. You don't have this. So what else can we use because we don't have this? I mean, when you go, hey, we don't have this. Can we order? Oh, you have this. It'll work. So you're always looking for things that will work if you don't have everything. And if you make changes to these kits, this polymerase chain reaction it's very simple. When you start making changes, things start working. So I've always found it's easier to ask someone else to do it because I would always just be like, oh, it doesn't matter. Let's do this. And you really can't logically think of why it doesn't matter. It does matter. I always found it easier to ask someone else to do it for me. Because I would always end up changing something. So I honestly think if you trained a monkey to do it, they would probably do a better job. Polymerase chain reaction. This is why I think it would be very helpful for you to know what DNA polymerase does. Because polymerase chain reaction takes advantage of what is DNA polymerase, how does it work, and how can we manipulate that to work to our advantage. DNA is replicated. So if we have a double strand of DNA, 5' end, 3' end, we have a complementary strand that is anti-parallel. How does DNA polymerase? So we have our DNA, this is our DNA to replicate. So in our solution, we have our DNA to replicate. We're going to add some things to it. We're going to add DNA polymerase. That's DNA polymerase. Now I'll make this a legend, a nice legend. So this is DNA. I'm going to put Taq polymerase. It's a polymerase that's frequently used. So Taq polymerase is a DNA polymerase. It's very, very, very stable at different temperatures. You're going to notice that we're going to be fluctuating temperatures frequently to melt DNA and to re-anneal DNA. Taq polymerase is a DNA polymerase that is resistant to changes in temperature, so it's frequently used. So we have our Taq polymerase. We have, I'll draw a few of these. These are our nucleotides. Our nucleotides, these are our, we're calling DNTPs. DNTPs are deoxynucleotides. These are our nucleoside triphosphates, and I told you if you don't like how I said nucleoside triphosphates, if you're like, no, it should be nucleotides. One, you're wrong. The two, I don't care. Call it whatever you want to call it. It doesn't matter. Our nucleotide triphosphates, if we're going to start replicating DNA, we need nucleotides. To add nucleotides. So our DNTPs are our nucleotides that we're going to add. And plus, we have to have primers. Primers, if you know anything about DNA polymerase and how DNA is replicated, DNA polymerase binds to single-stranded or double-stranded RNA or DNA. Tell me, single-stranded or double-stranded? It's kind of a trick question, isn't it? Because DNA polymerase, it binds to double-stranded DNA, but it doesn't replicate double-stranded DNA, right? It only replicates one side of DNA. So it unzippers your DNA. A RNA comes and binds to a specific spot, an origin sequence, to where your DNA polymerase can also bind. So it binds to double-stranded nucleic acid, right? One strand is an unzippered DNA strand. The other strand is a small segment of RNA that its only goal is to bind to a certain spot so that DNA polymerase can bind. It'll bind and it'll start replicating from there on out. You'll see it here in just a second. So why did we add DNA primers? We have DNA primers to mimic our something that we don't have in solution, which will be our RNA that will bind for DNA polymerase. This is to mimic our double-stranded binding site. In polymerase chain reaction, we will denature, we will melt. So we have heat, denaturation. So we're going to denature our enzyme, we're going to denature or melt our DNA. So we're left with two single strands of DNA. We'll mimic the unzippering normal DNA replication. We will unzip our DNA with heat. This is used. Did y'all use, when you did PCR, did you use a thermocycler? You didn't. A thermocycler is it's a really small instrument. You can put little tubes in, close it, and it cycles hot, cold, and you cycle it hot enough to melt DNA, then you can cycle it cold enough for a certain amount of time to re-anneal the DNA. So it cycles hot to melt, and then it will cycle cold slowly, because once we melt our DNA, we want to anneal our primers to our DNA, right? Because DNA polymerase is not going to replicate single-stranded DNA by itself. It has to have a double-stranded section to bind to. So we have our primers. This is the goal of our primers. So the goal of our primers is to bind to a certain spot where DNA polymerase can start its replication. DNA polymerase will recognize this sequence. So DNA polymerase will recognize this sequence and bind. So we have DNA polymerase binding to both of these sequences. And from this point, DNA polymerase can do its thing, replicate, and extend. So from one strand of DNA, denature one strand of DNA, replicate, and now we have two strands of that exact same DNA. Extend our DNA. And from one strand, we have two strands. One strand, we have two strands. And guess what you're going to do? You're going to do it again. And that's what your thermocycler is for. You're not going to do it again. You set up this reaction and you push a button, and your thermocycler is set up to do this over and over and over and over and over and over. Iteration after iteration after iteration. Most thermocycler, if you buy a kit, most instructions will have you do this 20 times. And they estimate that after 20 times, you will be left with an order of magnitude of 6 greater DNA. You can take small amounts of DNA. You can replicate this over and over and over again to get a large measurable quantity. It's used in forensics from a single strand of hair, from a flake of skin. You can take that DNA, replicate it over and over and over again to a degree in which it can be measured. It can be run through instruments. It can be seen. I don't do forensics. I don't want to say I don't care about forensics. I don't do it. What I do care about is protein purification, enzymes, studying enzyme mechanisms, and the exact same thing holds true for this. When you make a change to what do I want my bacteria to produce, and I say my bacteria, I want you to produce this protein so I can study it. I want to study this enzyme. Back in the day, I told you all the lowest hanging fruits, they're gone. All the easy enzymes to study, they're gone. Hemoglobin, protein. Extremely easy to study. Why? Because you can literally go to the butcher's market and buy meat, grind it up, and get blood, and study it that way. I mean, it's easy. All the low-hanging fruit's done. Now, the difficult stuff comes, and we have to use methods like this to amplify what we're looking at, what we're working with. You should understand the fundamental concepts of polymerase chain reaction. Understanding how DNA polymerase works makes this absolutely trivial. Because understanding how DNA polymerase works, you can see exactly why this makes sense. So I encourage you, if this doesn't make sense, please go back, look at exactly why how DNA polymerase works, and what's required for DNA polymerase to work. Once you see this, and once you understand it, this is going to make a lot of sense, and it's going to be absolutely trivial. Lastly, I want to tell you what is going to be on your exam. What do you need to know from these chapters? A lot of people have been asking, so I shall tell you. From these chapters, you need to know what does RNA do, specifically mRNA, RNA, tRNA. I said at the very beginning, those are things you need to know. How is DNA and RNA replicated? DNA and RNA polymerase, how do they work? Why do you need to know how DNA and RNA polymerase work? Because it makes polymerase chain reaction, understanding polymerase chain reaction much easier, and that's something you need to understand. So I will ask you questions about polymerase chain reaction. You need to know about base hydrolysis of DNA and RNA. Which one is more susceptible? Why is it more susceptible? And what are the possible products you would see from hydrolysis, DNA and RNA? You need to know how bases pair, both Watson-Crick and Hookstein. You should be able to identify either. Identify Watson-Crick base pairing, identify Hookstein base pairing. What are the ramifications of Hookstein base pairing? You should know the secondary structure of DNA and RNA. Secondary structure meaning what do inverted palindromes, what kind of ramifications do those have? As far as secondary structure, hairpins, cruciforms. You should know various secondary structures, anti-codon stem loops, RNAs, specifically tRNAs. You You should be able to identify the differences between B, Z, and A forms of DNA. And I don't want to know bonding. I don't want to know any of this kind of stuff. You know one is more prevalent than the others. You should be able to identify from a drawing any of these. To know how DNA is denatured, how DNA is annealed, via temperature or via pH, effects of tautomerization we talked about, effects of tautomerization, pH on the formation of a helical strand of DNA. You should know about the hyperchromic effect. I promise you will have a question about the hyperchromic effect. Most likely explaining the hyperchromic effect. You should know restriction enzymes, cloning vectors, how they are used. You should be able to identify various types of sticky end or blunt end cleavage from restriction enzymes. Now, there's a huge table in both of your books on all of these. You don't have to know those tables to answer the questions. If I asked you, I gave you 4 restriction enzymes and asked you which of those yielded blunt end cleavage, you don't have to know the table. All you have to be able to identify is from. I would actually give you one side of a cleavage site, and from there you would tell me. Don't memorize the table. That would be a very bad idea. From a plasmid, from a cloning vector, you should be able to identify ramifications of various restriction enzymes. If I asked you what happened if you cleaved at this spot or this spot or this spot, and I told you what type of, not really what type of cleavage it was, but if I gave you this information, you should be able to tell me ramifications on that. What am I disrupting? Am I disrupting A, B, or C from this? When you do your hormone column, so it'll be much easier, I promise. And lastly, you should be able to talk in detail. Yeah, you should be able to talk in detail about polymerase chain reaction. What are the reagents necessary? What do the specific reagents do? Why do we add DNTPs? Why do we add DNTPs?
They're building material.
It's a building material. How else are we going to have DNTPs if it's a different question if we're talking about inside a cell, right? We're not talking about inside a cell. We're talking about inside a test tube. So if you don't add these things, they're not there. So they're building materials. So if you should know the function and the roles of all of these and why do we have to add these? Because we're not doing things in a cell. We're trying to mimic what happens in a cell in a test tube over and over and over. So we have to add these things. We don't have any building materials in our test tube. Those are the things you should know for this, from these chapters for the next exam. This is chapter 10, 11, and 12. The next 2 classes we'll cover chapter 3. I specifically slowed down in this chapter a little bit because I want you before Friday to read chapter 3. That is your goal. Please, for the love of all who is good and sacred, read chapter 3. Read it before you come to class, not after you come to class. So one thing I've told people over and over and over again, I haven't done well the first 2 camps. I haven't done well. What can I do better? What can I do better? The one thing, read the chapter before you come to class. It makes such a difference. You have no idea. I would rather you do that than do homework. If you said, one, I'm going to read before I come to class, or I'm going to do homework, I would say read before you come to class. I would rather you do that than I'd rather do both. Right? Do both. I would rather you read. More important than doing homework, without a doubt. Read before you come to class. Read chapter 3 before Friday.